Nemerteans, or ribbon worms, are unsegmented and bilaterally symmetrical. When first examined, they are coelomate worms with separate mouth and anus.
Nemerteans consist of about 1,200 species of worms. Their lifespan has not been widely studied, but they are thought to be long-lived, surviving for a year or more. Some have a direct life cycle, others go through a complex metamorphosis.
Nemerteans can be found at all ocean depths, freshwater, and terrestrial habitats. Of the 1,200 species of Nemertea, most of them reside in a marine environment.
Most Nemerteans reproduce by sexual reproduction, being either male or female. However, some freshwater and terrestrial species can be self-fertilizing.
The gonads in Nemerteans are temporary structures only, developing during the breeding season and then degenerating again. Each ovary or testes has its own duct to the outside. In a very few species, the eggs are retained within the female and hatch as small copies of the adult to then be released into the world.
Normally, the eggs are laid in gelatinous strings and the sperm is released into the water near them so that fertilization takes place outside the female's body. The development of the eggs can be either direct or indirect. Direct development is where the eggs grow into a smaller version of the adult, as in humans. Indirect development is where the egg grows into a larvae, which then later undergoes metamorphosis to gain the adult form.
The feeding appendage is held in the sac above the worm's gut. When prey is detected, muscles rapidly contract and force fluid into the sac, which turns the proboscis inside out to catch the prey. It is toxic to the prey and paralyzes it. According to scientists, there are no known positive or negative effects of Nemerteans on humans.
Ribbon worms can glide over the surface by ciliary action aided by mucus secretion or move quickly by peristaltic contractions of their body musculature.
Here we have our phylogenetic trees. The first is based off of our data matrix and observations. The other is based on the 16S rRNA sequences. Both trees start off with puriferously outgroup, then the second branch separates into Cnidaria and a different phyla. In the 16S rRNA tree, Nemertea does not have a sister taxon, whereas in the data matrix tree, Nemertea's sister taxon is Annelida. Annelida is the closest relative to Nemertea in the rRNA tree, and Arthropoda is the closest relative in the data matrix tree. Both trees have a Echinodermata in the last clade, but with different sister taxons. One of the reasons why both trees are different could be when we introduced chordata, as we dissected fish and not mammals. Therefore, we examined gills as the respiratory characteristic instead of lungs. Another reason is human judgment, where we analyze certain characteristics with our own judgment, such as counting the nerve net and the nerve ring, different and not similar.